All right, this recording is for Biology 1406. This is the third lesson in Unit 4. The chapter is 16 out of the OpenStax biology book on gene expression regulation. All right, so regulation is required because every cell in an organism, and for single-celled organisms, that's just their one cell, of course, contains every single gene that they've got, and not all of those genes need to be expressed. In other words, an RNA product or a protein product actually produced all the time from a given gene. So, those things need to be produced when they're needed. So there has to be a mechanism of upregulating or downregulating, turning on or turning off expression of a gene to match the needs of the cell. So the reason we want to do that regulation is because we need to conserve energy. If a cell were to express every gene constantly, producing the products of that gene, it would be using energy to make things that may not be necessary and could possibly, therefore, not have enough energy to produce things that are needed, that are necessary at that particular moment. So that's one regulatory issue. Time. There are periods in a cell's functioning when certain genes need to be expressed and other times when they are not. So they are time sensitive. For instance, genes coding for proteins that are only used during cell division would only appropriately be utilized at that time. So that's a time component to regulation right there. And of course, space. The cell can't store an infinite amount of supplies without getting infinitely larger, and we know that cell size is limited both in prokaryotic cells and by way of their increased internal complexity, a much larger but still finitely large cell is possible in eukaryotic organisms for still the same reasons. They don't have an infinite ability to store resources and products. All right, another thing that is really important, and this is really characteristic of multicellular organisms. So when we say a somatic cell on this slide right here, a somatic cell refers to a cell of the body of a multicellular organism. And a somatic cells specifically exclude gametes and gamete-producing cells, which are a different group of cells. They all contain the same genetic content, with very rare exception. Every cell in the body of every animal, every plant, every multicellular fungus contains the same genetic information, but different cells with different functions. Like here we're looking at an eye, a vertebrate eye, which is an organ made out of lots and lots of different tissues. And here we're looking at organs of the vertebrate digestive system, the liver, the gallbladder right here, bile duct, the pancreas, and the duodenum of the small intestines. All are examples of organs constructed of different tissues. And those different tissues refer to cells that have expressed different parts of their genome, different portions of their genetic content, to have particular features, structures, and capabilities that are unique to the cells in that particular organ, whatever it may be. All right. So some general models. We'll take a look at transcription in prokaryotes and eukaryotes right here, and then we'll look in a bit more detail. So here we've got an illustration of just a very simple schematic of a prokaryotic cell. So we've got the cell membrane. Recognize there's almost surely a cell wall outside of this, but it's not important for this conversation. The cell contains cytoplasm, which is the cytosol plus everything larger molecular structure suspended in it. As you should know, a prokaryotic cell typically has one circular chromosome. There are exceptions, and there's also auxiliary bits of DNA that can be present, but those are, again, not particularly important in this conversation. Key thing is transcription and translation, which we've already studied, can occur and does occur pretty sequentially and in rapid succession and semi-simultaneously. Transcription must start occurring first. We can't have any translation until we have a transcription product. But even before transcription is finished, ribosomes can attach to the transcription product, the messenger RNA, and start synthesizing, the relevant polypeptide. So in this particular drawing right here, one genetic section has been transcribed or is being transcribed. It's not finished yet because we still have them intertwined right here with the messenger RNA hanging off into the cytoplasm. And this ribosome system right here attached, or the drawing is really backwards. They should be running in the other direction, but they got that a little bit wrong. So a ribosome is attached right here and started working its way down, along the messenger RNA, synthesizing a polypeptide chain, and more of those are latching on behind and repeating the process. These are done backwards. They missed it on this drawing right here. This should be the longest polypeptide chain, and this should be the shortest one because they're going to be reading from the 5' end of the messenger RNA and working down it in the 3' direction. Translation occurs reading the messenger RNA 5 to 3. All right. By comparison, a eukaryotic cell, amongst all the other differences, is we have a nucleus containing the nuclear material, as opposed to the nucleoid in a prokaryotic cell, which is just a lot of proteins that structurally support and regulate the chromosome, but they don't define a separate compartment. So the only compartment is the actual cell compartment, the cell membrane. In our eukaryotic cell, we've got the nuclear membrane shown right here with the nuclear pores going through it, and recognize this is actually a double membrane. So this would fold around and make the outer membrane, which includes the extensions called the endoplasmic reticulum. Again, not important in this particular picture. And here we are in the cytoplasm of the cell, and the cell membrane is going to be somewhere further outside of the picture right here. And the point to make right here is that transcription and translation are separate processes that occur in separate compartments. Transcription occurs, as in the prokaryotic cell, right there where the DNA is. After we've transcribed that pre-messenger RNA, there are going to be the 3 types of editing that occur. The addition of a 5' cap, the poly A tail right here, and then the excision of, it looks like in green in this drawing right here, of introns, and the combining together of what's left, color-coded blue right here, the exons combined together. And that is what's exported from the nucleus, and then we can have ribosomes attached to it and start transcribing. And like in prokaryotic cells, there's no rule that says more than one ribosome system can't attach to a given piece of messenger RNA and start transcribing them. And again, translation, start translating them. Translation should be occurring from the 5 to the 3 direction, so this should really be the longest chain, and this should be the shortest one. They've got their picture backwards right here. As usual, I've borrowed this material and edited it, but I'm not editing these photos. That's a lot of work. Unless they're screamingly bad, I'll see if I can fix these sometime. But I'm using this material provided by the publisher. Gene expression in eukaryotic cells is regulated importantly in several different ways. Epigenetically, gene regulation occurs by controlling which pieces of DNA are actually accessible for transcription. All right? So that can be controlled. We don't change the DNA code, but we change whether it can be read. If it can be read, we can transcribe it. If it cannot be read, we can't transcribe it. Then we can regulate the process of transcription. Does it occur? We can regulate stages in this modification after transcription right here. That's post-transcriptional modification. We can regulate the process of translation. Does it occur frequently? Does it occur rarely? And then the product of translation, the polypeptide right here, can be modified, typically in the endoplasmic reticulum system, the endomembrane system of the eukaryotic cell. So we have a number of ways in which we can regulate gene expression. Right at the end, where the proteins are being used, or before we even get to the translation stage. That's epigenetic control. Epi means outside of genetic, so outside of the gene regulation. In comparison, regulation of gene expression in prokaryotic cells is almost completely restricted to regulating the act of transcription. In other words, how and when does RNA polymerase attach to a gene and initiate the process of transcription? When that occurs, that is the main regulatory mechanism these cells use. So, kind of stepping back and looking at the two of them for a minute right here, what you've got is a simple cell with one highly regulated way in which it controls gene expression. And the important point I want to show is that simplicity is really the driving rule with prokaryotic cells, both in their structure, in their organization, and in their regulation. And that's part of the package of being a small, very, very, sort of minimum required materials sort of cell. So you can think of prokaryotic cells, bacterial cells, as, and there's a lot of range in this still, but as sort of the minimum viable, functioning, self-reproducing organism possible. It's really hard to get too much smaller than a bacterial cell, or have much less information than you find on the genomes of a given bacterial cell, and still have a cell that can function autonomously and live in the environment and reproduce, and all of that sort of stuff. But simplicity and efficiency are the driving factors in expression in prokaryotes. Eukaryotic cells, complexity and sophisticated regulation and responsiveness, lots and lots of variation and responsiveness are the hallmarks of eukaryotic cells. Because they are aerobic, they produce enough ATP to power, they've got excess energy available to power all the complexities, but they burn quite a bit more resources. They use up lots more ATP. That's why they've got to have much more complicated cellular metabolisms, cellular respiration pathways and all of that. And because of that extra power and complexity, they're able to do a lot more, finessing with their regulation of gene expression. And there's some efficiencies built in right here. If you regulate epigenetically, you waste the least amount of energy because you don't even open up DNA to read it. Saving ATP in every step along the way, energy in every step along the way. On the flip side, if you regulate your proteins at the post-translational level, you have the ability to most accurately modify your protein products to the environment in real time that they're being exposed to. That is a important idea as well. All right, so just to sum up some differences between prokaryotes and eukaryotes that you want to know. Of course, the first bit, pretty well understood by now, presence versus lack of a nucleus. And what this means is that, the DNA can be in a different regulatory environment in the nucleus than a prokaryotic cells can, because it's in the cytoplasm. And therefore, this becomes true as well. RNA transcription and protein formation occur quite overlapping, almost simultaneously. Obviously, transcription has to start first, but protein translation can occur even as transcription is still proceeding. Whereas in eukaryotic cells, those two processes are separated in time and in space. Regulation is relatively straightforward in the sense that almost all of it occurs at the transcriptional level. Do we or do we not transcribe the gene? Whereas regulation in eukaryotic cells occurs at many different levels for many different reasons with different efficiencies. Energetically efficient, responsiveness is maximized if we regulate at the post-translational level. We've got the protein there, but we determine how long we use the protein and how we use the protein and how we modify the protein. That's what this is all about. And epigenetic regulation means, do we even look at the gene or not? Do we even have access to it to express it? That is a non-issue for a single-celled organism, but for a multicellular organism, that's a huge deal. What we want to do is turn off the genes that code for characteristics that a cell of a particular tissue is never going to use. Alrighty. A very important idea in prokaryotic DNA organization is the idea of an operon. All right. And so operons are a group of genes having to do with the same biochemical pathway that are organized, as it says here, as a block, and all regulated by the same promoter region. And then those operons can be repressible operators or activators, and there's inducers. There's these proteins that interact with the repression or activation pathways. We'll talk about that in just a tad. But the idea is that that operon can be upregulated, which is an activating pathway, or downregulated, that's a repressing pathway right here. But an operon consists of a bunch of genes all controlled by the same promoter region. So let's look at a picture of that. So here's a promoter, and remember that a promoter is the region where a RNA polymerase plus a bunch of transcription factors attach to this chunk of DNA. A promoter is some DNA, just like all of this is DNA. And then we've got some more of, it's the extension of the promoter called an operator, which just means a region where certain molecules, additional molecules can attach that determine whether RNA polymerase can pass through or not. And then the actual genes that code for distinct protein products. And these are all strung together in a group right here. So this is an example of an operon. One promoter, and then in this case, 5 genes all in a row. So far, what we've looked at is the idea that a gene, like TRPE right here, has a promoter. And by extension, the idea is that each of these other genes has a promoter as well. Well, in prokaryotic cells, quite normally, groups of related genes are strung together, one after another, and all controlled with one promoter. The idea being, if I need this, I'm going to need this, 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 and this as well, all 5 of these. If I need one of them, I need all 5. And so let's go ahead and transcribe them simultaneously. Right? That's an efficiency. What it doesn't allow for is just expressing this gene without expressing the other 4. That's not really possible in this situation. So prokaryotic cells don't have that ability. All right, so this, in addition, we're looking at a repressible operon. All right, so repressible means that the default setting, if you exercise the least control over this at the cellular level, the default setting is that this operon gets expressed. RNA polymerase is likely to attach, pass through the operator, and transcribe all of the genes in the operon. That's what we mean by a repressible system. It can be shut off, but if you stop shutting it off, it automatically comes back on. Right? That would be like a door that naturally stays open. You can close it, but if you don't keep it closed, it just pops back open again and is expressing the all right, so that's the idea of a repressible operon. And let's go ahead and take a look at it in the active state, so non-repressed. The repressor molecule is not attached to the operator right here. So once RNA polymerase was attached to the promoter, remember Tata boxes and transcription factors, a bunch of different proteins here, that say, hey, we need this. It goes ahead and runs on through and is going to transcribe all of these genes. So what we'll get is a messenger RNA coming off of here that has a start codon somewhere along the way here to begin translating into a protein, a stop codon here. There'll be another start codon here, another stop. So even though this is one piece of messenger RNA, it'll have 5 different spots that can be translated by ribosomes. So, it'll produce 5 discrete proteins. So a very nifty little system. And so the idea here is when there's a lot of tryptophan available, tryptophan is an amino acid, and this is a pathway for converting one amino acid into another amino acid, the tryptophan pathway in particular, because we know it really well. And the idea is that when tryptophan is really, really abundant, it's in high concentration in the cytoplasm and has a high probability of being available, and it attaches to an active site on this enzymatic protein called a repressor that is The repressor is a protein produced by another gene somewhere else. In the case of this cell, it's in another location on the chromosome. And this protein here has a couple of different states that it can exist in depending on what's around. So if tryptophan attaches to it, it takes on a shape that allows it to bind to the operator and block transcription. So when we've got lots of tryptophan, there's no reason to go ahead and make more of it. We'll save energy. But if tryptophan becomes rare, there's a low probability of it being around to attach to this repressor. And so the repressor changes to a shape that can't stay attached to the operator. And now we can go ahead and express this gene. So that's what we mean by repressible. It tends, and this is regulated by a negative feedback process. Too much of the product of this metabolic pathway, not too much of it, but lots and lots of that product inhibits production of the product. That's negative feedback via this repressor protein. All right, let's go ahead and take a look at this animation.
Tryptophan operon, TRIP operon for short, is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The TRIP operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRIP-R, encodes a dimeric repressor protein on a slow but ongoing basis.
Dimeric means that the protein has 2 different shapes. You may recall the term chimera, which is a Greek term representing an animal that is made out of pieces of lots of different animals. So kind of, a related term. Dimeric meaning having 2 forms or 2 shapes.
And the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the TRIP operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRIP-L, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRIP synthesis pathway. TRIP-E and D for anthranilate synthase, TRIP-C for indole glycerol phosphate synthase, and TRIP-B and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRIP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes.
So that's that dimeric state. Here it is without tryptophan attached to it, so it has one shape. With tryptophan attached to it, it has a different shape that allows it to actually bind to DNA. It's going to bind to the, I believe, the major groove of the helix in that location.
Thus, tryptophan is said to be a co-repressor. Like the LAC repressor, the TRIP repressor does not stick like glue, but periodically dissociates, allowing a low, basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter 2 tryptophan codons in a region called 1. They wait for TRIP-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the TRIP tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
All right, so that was quite a bit more information than we needed right there. The big thing the things that for your test that you want to make sure you can talk about are the difference between the promoter and the structural genes, what that actually means, and all of, promoter plus a bunch of structural genes is an operon, and then how that repressor can go from the inactive state to the active state based on the presence or absence of tryptophan. All right, let's look ahead. Now we're going to look at a inducible operon. So this is the lac operon, lac for lactase, and this is for metabolizing lactose, the enzyme lactose, or sorry, metabolizing the disaccharide lactose. This is familiar to us because humans, of course, some of us can metabolize lactose. We're lactose tolerant, others cannot, we're lactose intolerant, and that has to do with gene expression. Do we express the genes required to metabolize lactose? We, being eukaryotic organisms, don't have those genes organized into an operon. Instead, each of the, we've got our version of these genes as well, but they're in different locations, potentially on different chromosomes, each with their own promoter and operator in front of this one, promoter and operator in front of that one, and this one, so that we can regulate each one kind of independently. But, we do the same thing. But here we're looking at an operon in a bacterial cell. And this one's inducible, which means the natural state is off, but under certain circumstances, we can go ahead and transcribe and translate and produce the enzymes for lactose metabolism. If glucose becomes rare inside a cell, we start having larger and larger amounts of ADP, adenosine diphosphate, and if we're really short on glucose, even high concentrations of AMP, adenosine monophosphate. And as those adenosine monophosphate gets converted into a cyclical molecule, which is a signal. It's an internal, it's not a hormone because it's not in the blood, but it's a signal molecule that powers up all kinds of actions inside a cell, and that includes binding to what's called a cap protein, and that is a positive regulator that in its active state binds to the promoter region right here and induces RNA polymerase to attach and really transcribe this thing very, very rapidly. So if the cap is attached because of a shortage of glucose and therefore a whole lot of AMP in a cell and not much ATP, this complex will appear because the two join together, AMP plus this, and bind to the promoter region right here and make it more likely that RNA polymerase will attach and then transcribe this entire operon. That's what's meant by the great big arrow here. In the absence of that cap system, this operon is only promoted rather sluggishly at best, and that's what's meant by the smaller arrow right here. All right. So the idea here is that cells will normally use glucose as their power source, and if they can get it some other way, they're not going to express much of the metabolic equipment that's required to metabolize other sugars, because those other sugars are more expensive to use and don't pay off as well as glucose. But if glucose is absent, then the cell needs to switch to using other sugars, and that's what's going on here with that cap-camp complex right here. All right. Additionally, there's a repressor at work right here, and the repressor, very much like the previous example with the trip operon, is a dimeric protein, so it has two different shapes. The active state, where it's bound to the operator, blocking it so that RNA polymerase can't push on through, and the inactive state, where it doesn't attach to the operator, and therefore the operon can be transcribed. In this particular case, the relationship between the metabolite and the repressor is reversed. Tryptophan, when it was abundant, attached to its repressor and caused the repressor to attach to the operator. Lactose, when it's present, when it binds to the repressor, keeps the repressor from attaching to the operator. So we have it out here in the cytoplasm. The absence of lactose allows this repressor, it has a different shape, to attach to its operator. And so that's the other part of the question. So do we transcribe the lac operon? First answer, well, if we're short on other sugars, yes, let's go ahead and activate it. Okay. B, can we transcribe the lac operon? Well, the only point of using it specifically is if lactose is available. So if there's a lot of lactose floating around, then lactose can be used as a stimulant to activate this particular gene right here. So this process right here, the CAP plus CAP complex right here, is going to activate the lactose pathway. It's going to activate the starch pathway. It's going to activate the maltose pathway. All of the other complicated carbohydrate sources that this particular bacterial cell can use will be activated by this system. But then, specifically, can we use lactose? Is only useful if the lactose is present. So the repressor also goes in right here. So if this isn't happening, if there's no lactose around, the repressor still sits here, and it will still be sitting here in this location right here. And even though we've got CAP and CAP all combined together, RNA polymerase will get stuck at the operator because the lac repressor is in its active state. Alrighty. So, and here, this is telling exactly that story. So, provided the repressor is not around, and that's because lactose is attached to it, and CAP have attached right here, we're going to vigorously transcribe this operon. If lactose is not present, the repressor does attach right here, and even though the cell is starving and short on fuel, there's still no point burning up ATP, making these enzymes to break down lactose when there's no lactose available. That wouldn't make any sense, so that's why this works. All right, let's go ahead and take a look at that animation.
The lac operon is a bacterial system for regulating genes that control the utilization of a disaccharide, lactose. The operon controls expression of genes for beta-galactosidase, which splits the disaccharide, permease, which transports lactose into the cell, and transacetylase. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. When there is no lactose in the cell's environment, it would be wasteful of the cell to make these proteins. The cell keeps the RNA polymerase binding site turned off. Let's see how. Upstream from the lac operon is a regulatory gene, LAC-I, which encodes a repressor protein. The repressor tetramer is slowly but continuously produced by the cell. Now let's look at the control sequences, the promoter and operator, for the structural genes. The operator overlaps the promoter. If a repressor molecule is present, it can bind the lac operator. It doesn't stick to the DNA like glue, but continually binds and releases, so the operator is not bound by the repressor at all times, and structural genes are transcribed at low or basal levels. When there is little lactose present, there is no reason to waste energy making the enzymes to process it. But what happens when there is a lot of lactose to process and the cell must use it as its carbon source? The repressor proteins have sites that bind lactose. Lactose binding causes a change in the shape of the repressor so that it can no longer bind the operator. Without competition for the promoter, RNA polymerase can repeatedly transcribe the structural genes, and translation of the now abundant mRNAs makes the enzyme for processing lactose. The lac operon is an inducible system, and lactose is its inducer.
All right. So that's inducible and repressible gene regulation. Now, please be aware, we showed you these processes at work on an operon, They're at work in a eukaryotic cell as well. The difference is there's no operon. Each of the structural genes will be controlled by its own promoter and operator region, and those might have slight differences in them so that they can be regulated independently of each other, and that's the strength of the eukaryotic cell. So it can make one of the structural proteins, one of the structural genes can be used to code for one of the proteins needed without the other three automatically being transcribed and translated at the same time. So we can individually regulate each individual gene in the lac pathway. Eukaryotic organisms, like all mammals, have a lac pathway because they can digest lactose at least as babies, as infants. But then that pathway gets shut off, permanently repressed. Permanently shut off, no longer induced in adulthood normally. So there's an example, but each gene would be independently regulated rather than the operon controlled as a unit as it is in a prokaryote. Now, going over and looking at eukaryotic only gene regulation issues, remember that we have a number of different places in which we can regulate gene expression, starting with epigenetics, which is to say around the genes. All right. So epigenetic regulation occurs before transcription. All right. All right, let's take a look at the human genome for just a little bit and remind ourselves of a couple of features. And this is true as a generalization for all eukaryotic cells, although the numerical bits are distinct. So in a human, you've got about 20,000 genes spread out over 23 chromosomes, 23 pairs of chromosomes, so 46 in total. So a bunch of genes per chromosome. And these 20,000 genes code for about 100,000 proteins. You should also remember that our DNA is wound around these histone protein complexes, and that to express a gene, we have to unwind that DNA, separate the histones apart from each other in order to get at the DNA to express it, for RNA polymerase to be able to get in there and attach to it. All right. So here we're looking at an example of a DNA double helix wrapping twice around a histone complex, which consists of 1, 2, 3, 4, 5, 6, 7, 8 proteins. And here's a photomicrograph of some DNA in the nucleus of the cell, and the little dots we're looking at here each represents a histone complex. So we're seeing really the histone complexes and just barely getting a hint of the DNA molecule itself threaded along between them. So we can pack and unpack DNA by binding or releasing the histone groups from each other. So this is the same histone set of 8 proteins. Here's another one, and there's space in between so we can get at the DNA right here. Here it's all packed up tight, and you can't get in here to read the DNA at all, anything in this wound up area here. And this is what DNA looks like when it's compacted in preparation for cell division, that condensed chromosome that we've talked about before. And how does that happen? Well, on the histone tails right here, we've got histone, these little tails sticking off of our histone complexes right here. And if we add acetyl groups to those histone tails, it's called acetylation, to add an acetyl group. Remember, an acetyl group is a CH3 functional group that you can make, add to a organic molecule to make basically a longer carbon skeleton. If we add those acetyl groups, we block off a charged area that normally caused this histone to attach to that histone over there, which was the case right here. So when we acetylate the histones, we unpack DNA. Now, in addition, we can add methyl groups primarily to the DNA itself right here, although there's some with, the histone proteins as well. And methyl groups attached, the methyl group is the CH3. Sorry about that. The acetyl group is not. The methyl group is the CH3 addition right here. When we attach those methyl groups right here to the DNA and, and it's to the protein, the backbone of the DNA molecule, we induce packing right here. So methylation packs, acetylation unpacks. Now, these tags modify the expressibility of DNA, but they don't change the DNA sequence themselves. It doesn't change any of the lettering or the meaning of any of the lettering. All it does is makes the DNA more or less accessible. So that's why cells that produce the lens in your eye don't express any of the genes involved in skeletal muscle function, or nerve impulse propagation, or production of digestive enzymes. Those genes are off, and so you can bet they're all going to be methylated, and they're going to be lacking acetyl groups in that area, so that the DNA remains packed and non-expressible for the duration, the lifetime of that cell. Because the cells that make up the lens of your eye grow up quickly, produce a huge bunch of crystallin, which is their transparent protein they make, and then die at maturity, which is why the lens of your eye is immunologically neutral. It's not alive and can be transplanted. It has no living cell components. Anyway, a little bit about that. All right, so that's what we mean by epigenetic regulation. So here's an illustration of that process at work right here. We've acetylated DNA right here, so we're exposing, components right here. We're adding methyl groups right here, which regulate whether we can express a gene or not. So if you add methyl groups, you downregulate expression of a gene. If you acetylate histone proteins, you upregulate expression of a gene. Let's go ahead and take a look at this animation right here.
Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation.
So the idea here, it said heritable. So methylation and acetylation are both heritable, and what that means is you're developing as an embryo. And you've got a bunch of regular old cells, in the future eye that are going to ultimately be the parent cells for all the cells that make up the lens of your eye. And at some point along the way, those stem cells, which are just rapidly dividing cells that can make any part of your body, are going to differentiate into lens-only producing cells. They will stop being stem cells at that same time. And the way they turn into lens-only producing cells is by methylating the genes having to do with anything that has nothing to do with making the lens proteins, crystallin proteins, and the few other things that are needed to keep the cells alive while they produce that, and not acetylating those regions right there. And that pattern is heritable. So once that's occurred to a stem cell, when it divides, it replicates that pattern in the daughter cell. And when those cells divide, they replicate that pattern in their daughter cells. And so they may still be stem cells in the sense that they can divide indefinitely, but they've become tissue-specific cells. They can no longer go backwards and be capable of producing all proteins. They can only produce the ones necessary for the particular tissue that they are now. That's what we mean by the heritable part right here.
Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made.
So you don't need to worry about the hat or, the various molecules that are involved in adding or removing the acetyl groups. That's kind of beyond our requirement. You'll see it's not in the study guide, but the presence or absence of the acetyl groups themselves is. And so, nothing happens just out of thin air. There's always something causing everything to happen. So of course there's some enzyme somewhere that is busy acetylating and deacetylating. But in this illustration right here, you saw the DNA helix unwind from the histone complex, and that makes it much more accessible to the transcription factors and to RNA polymerase that would induce transcription.
Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
Alrighty. So remember that, and this is particularly true in eukaryotes, we're going to have a bunch of different transcription factors present to induce RNA polymerase to attach and start transcribing the DNA and producing a RNA transcript. Let's go ahead and take a look at this animation here.
Our transcription animation provides an overview of the major steps involved in RNA production. In this animation, we will take a closer look at the events that prepare the DNA for transcription. mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription.
Where do these transcription factors come from? Some come from the cell itself. This given cell produces a protein that ends up being a transcription factor, so it's kind of auto-regulates itself. A lot of other times the transcription factor is the product of a transduction of a hormone, of a signal from somewhere else in the organism from other cells that induces this cell to go ahead and produce a protein. There's plenty of that going on in you all over the place.
These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed.
In this drawing right here, represents an ATP or maybe a GTP, guanosine or adenosine triphosphate. And so here is a phosphorylated protein that has been energized so that it can bind to another protein and cause something to happen. Very characteristic of something we call signal transduction, the movement of information through a cell.
The most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription.
All right, so let's finish this off right here. So, remember again, our promoter is a region upstream of the coding sequence. All right, that can be quite some distance away from the, that can be, a moderately long piece of DNA right here. Remember that we have that T and A rich region within a promoter called the Tata box, which is just upstream of the transcription start point. So that indicates where RNA polymerase attaches. And so we have these distal control elements, which just means really far away. We have the promoter immediately upstream of the structural gene. And this is a eukaryote, so there's only one gene. There's not a string of genes here, no operons in eukaryotic cells. A bunch of transcription factors. When all of them are all together right here, then we can go ahead and transcribe. RNA polymerase attaches, and we can start the transcription process. All right. So there's a number of different things that go into this, and I'm not going to be asking you about distal control elements or mediator proteins, but do remember transcription factors. That's just a grab bag for all of the various, typically proteins. They can be other molecules as well, but typically they're proteins that collaborate to regulate expression by regulating the ability of RNA polymerase to attach and to proceed down, transcribing the structural gene. All right. So we do remember the post-transcriptional modifications that can occur in eukaryotic cells in the pre-messenger RNA. And those modifications, one of them is to cut out introns. And the model we showed you before was that we would have, an exon, intron, some pattern of those. And the idea is that those introns are all cut, and what's left is spliced together. 1, 2, 3, 1. And here we've got a slightly different version, 1, 3, 4 right here. And we'll explain that in a minute. But that's the intron-exon processing right here. All right. Importantly, in different cells in use, skeletal muscle versus smooth muscle cell, the same gene, the cutting and splicing pattern, can be different to create two final structural messenger RNAs that code for final structural proteins that would necessarily be different from each other. That's what this represents. This is different from this. They're similar, they share pieces, but they're not identical. So here's some extended examples right here. So for a given sequence of exons and introns, gray for introns, colored for exons right here, we could express just the first and the last one right here, and everything else gets cut out as an intron in some cell, including this that we're calling an exon in a different cell. So in this cell, the red area is understood to be, is recognized as a, in this cell, whatever cell this is in your body, the red region is recognized as an intron cut out and left inside the nucleus. And this is all that's produced. This is the messenger RNA and therefore the final protein. Conversely, we can get this pattern. And so we've got some other, variations on the same idea. All of these are really meant to illustrate the ways in which we can splice different bits. Whatever we splice together and export from the nucleus by definition is an exon. It doesn't have an identity until it makes it out of the nucleus. But the alternative cutting and pasting of bits and pieces of the pre-messenger RNA together gives us the potential to make lots of different proteins from the same gene. And that is very characteristic of eukaryotic cells and what makes different tissues possible. Very important idea. So, how could this evolve? Well, errors in the editing process, and perhaps the very first intron splicing events where a mutant aberrant bit of RNA that was inducing that process. But again, like any error in any process inside a cell, if the error turns out to be useful, in other words, makes something happen better than it did before, or makes something happen that's new and helps that cell function, that error is considered to be, beneficial, and selection, natural selection, tends to favor it. And so it accumulates. And clearly, the idea, the mechanism for alternatively cutting and pasting together bits and pieces of messenger RNA in eukaryotic cells has, on balance, turned out to be a very useful thing because it's extraordinarily common and abundant and highly varied from one organism to another, and it has a lot to do with what the differences are between particularly closely related species. Why are they different? Well, they probably have pretty much the same genomes with the same genes coding for the same proteins, but it is a little bit of epigenetic regulation and alternative splicing that has a lot to do with the differences between one species and another closely related species. Amongst the great apes, the differences between us and chimpanzees, etc., etc. All right. RNA itself, once it has been produced, and here it is, we've got the 5' cap, that's one of our 3 modifications. We've cut out whatever the introns are and left the exons preserved and bound together. We've put a poly A tail on here. Here is the final, messenger RNA molecule, and it's got this untranslated region at the beginning and this untranslated region at the end right here. The untranslated region at the beginning determines where this protein will go in the cell, and that is one of those 3 destinations I've asked you to remember. That we can use messenger RNA to code for a protein that stays in the cytoplasm. How does the cell know to do that? Well, it's the code in this, the U's, G's, C's, and A's in this sequence right here that tell this messenger RNA, or tell proteins what to do with this messenger RNA, where to attach the ribosome. Will it occur in a free cytoplasmic ribosome, or is it going to be over on the endoplasmic reticulum? And similarly, a different pattern here in the UTR determines whether we attach to the endoplasmic reticulum. That's what we mean by messenger RNA localization and translation. Is it going to be in the lumen of the ER? That'll be written in this UTR region right here. Or is it going to be in the on the membrane, and that'll also be here. The untranslated region in the 3' end right here codes for how many adenine residues are added on to make this poly A tail, and the length of this tail determines how long this messenger RNA will last in the cytoplasm. So we can control where it goes, ER system or cytoplasm, and we can control how long it lasts, poly A tail. 5' cap, remember, is the passport out of the nucleus into the cytoplasm. No cap, no messenger RNA. I mean, it would exist, but it can't get out of the nucleus, so it doesn't really take on its role as a messenger. All right. This is a little bit more than we need to know at this point. I'm not going to ask you about these for the record. Small bits of RNA, and there's lots of different types of small bits of RNA. So that means things that are transcribed. So we get these bits of RNA, but the transcriptions don't become don't end up being messenger RNA themselves. They just are little bits of RNA, and they can dramatically interact in gene expression as well. They're a very important thing, but that's for an advanced course. So we'll leave those guys behind right there along with this material here. All right. Okay. Last thing, once we make a protein, its duration in the cytoplasm is also regulated. So here we've got a protein, and as it sits, in the cytoplasm, doing whatever it does, it's an enzymatic protein carrying on some function. Over time, it gets tagged with these ubiquitin proteins. In the last recorded lecture, I talked about parked dead cars sitting on the side of the highway and how you see them accumulate these stickers, a green sticker, an orange sticker, and finally a red sticker, which is when the tow people know to come and get that thing and tow it away, for the police. And, that's what these do. So as these accumulate, as we accumulate these ubiquitin tags, when we get enough of them, a protein complex called a proteasome surrounds this enzymatic protein and digests it, breaks it all apart back into its raw amino acids, and so the whole thing can be reused. And the idea is that no protein is going to last forever in the cytoplasm. They all get recycled. And that's why we always have to make new ones to replace them. The reason we do this is because this could misfold. If it misfolds in the wrong shape, it won't do what it's supposed to do, and it may do something else that we don't want to have happen inside the cell. Shape is everything in proteins. So they're degraded preemptively before anything bad happens. That's the idea of ubiquitinization, adding ubiquitin tags to all proteins in the cytoplasm. So just to kind of wrap it up right here, a lot of disease, a lot of malfunctions in humans can be in any living organism can be the result of altered gene expression. Cancer, for example, is, as it says right here, not a single disease, but many diseases, in the sense that it is mutations or errors in the regulation of gene expression in a set of cells that, amongst the many errors, also allows for rapid reproduction of that cell in error. And so we get a tumor, which is a mass of tissue accumulating in some location. That tissue may or may not carry on its original function. Typically in cancers, the original function is lost. The cells cease doing what they're supposed to be doing, like synthesizing some, milk, the molecules of milk here in this breast cancer mammary gland right here. But instead, all they do is just reproduce themselves. If those cells stick together really tightly in a mass, that'll be a benign tumor. And the reason it becomes dangerous is because it grows larger and larger and larger and becomes physically debilitating in some way, or it blocks passage of, key fluids from some location to another. In the case of a breast cancer, a benign breast cancer is going to be disfiguring. And, if it grows large enough, causes complications, but be rather easy to take care of. You just surgically excise it. A malignant cancer is a cancer where the cells in the cancer metastasize, and that means they let go of each other for whatever reasons. They don't stay attached to each other, and so this dividing, non-specific cell can be taken away in interstitial fluid via the lymphatic system, so classically the lymph nodes get involved, or the circulatory system, and distributed to other parts of the body. Wherever that cell lodges and can still get at nutrients, then it will continue dividing. And so then we have cancers appearing in multiple locations, blocking function in lots of different places. And those are the more scary and dangerous cancers. So just as a summary, when we're looking at eukaryotic cells, we've got regulation before we even get to transcription. That's epigenetic regulation. We can regulate, just like in prokaryotic cells, the transcription process itself. After transcription, we're going to make those modifications to the transcription product. That's, splicing introns and exons, poly A tails and 5' caps. We have translation. We can regulate how long we translate by how long, that messenger RNA lasts. Short poly A tail, we're only going to translate it for a short period of time. Long poly A tail, we'll translate it for a long time. And then post-translation, the protein itself. How long does that last in the cytoplasm? And that varies and can be upregulated and downregulated. All right. And so cancer is just what's happening in cancer is that one or more of these processes involving genes having to do with cell identity and cell reproduction, most importantly, regulation is lost at one or more of these levels right here. So for instance, in cancers, classically, there's a tumor suppressor gene, P53, right here, which is a transcription factor in the cell cycle. So it induces the production of the cyclins that are involved in the cell cycle and cell duplication. And, a mutation in that suppressor gene right there is indicated, as it says right here, in the bulk of, in more than half of all cancer types. All right. I think that's about everything we want to talk about in this particular lesson. So that will conclude the lesson on regulation of gene expression.